Think about the plastic tip on the end of your shoelace. That little piece, an aglet, stops the entire lace from unraveling into a useless fuzz. Your DNA has a similar problem. Every one of your 46 chromosomes is a long, linear string of information, and the ends are vulnerable. These ends are called telomeres, and without their own version of an aglet, the fundamental process of life, of making new cells, would shred your genetic code.
Every time a cell divides, it has to copy its entire library of DNA. The molecular machine that does this, DNA polymerase, is incredibly good at its job, but it has one strange limitation. It can't start copying from a dead end. It needs a running start. For one of the two DNA strands, the lagging strand, this means it copies in short, backward bursts. When it gets to the very end, there's no place for that final little burst to start. So with every single division, a tiny piece of the end is left uncopied. The chromosome gets shorter.
Some cells have a way around this. An enzyme called telomerase can act like a construction crew, adding back those lost bits of DNA sequence. This is why embryonic stem cells can divide almost indefinitely. It's a kind of cellular fountain of youth. But most of your adult cells have turned their telomerase off. Why? Because a cell that can divide forever is dangerous. That's one of the definitions of cancer. So this shortening, this clock ticking down, is a safety feature. But telomerase is only half the story. The shortening is the slow threat. There's a much more immediate danger. Imagine you're a repair system inside the cell. You have one job: find broken DNA and fix it. A double-stranded break in the middle of a chromosome is an emergency. What does the natural end of a chromosome look like to this system? It looks exactly like a break. It's a dead end. Your cell's emergency response machinery wants to grab this broken end and glue it to something else, perhaps the end of another chromosome. This is called non-homologous end joining, and it would be catastrophic. So, how does the cell tell the difference? How does it flag the true ends as safe and not broken? It doesn't use a flag. It uses a disguise. It assembles a microscopic protein machine, a complex called shelterin. Think of it as a multi-part cap that fits perfectly onto the telomere. It has key components, proteins with names like TRF1, TRF2, and POT1. Their collective job is not to repair the end, but to hide it, to make it completely invisible to the cell's overzealous repair crews. Shelterin's method of hiding is an act of physical origami at a molecular scale. It doesn't just sit on the end. A protein called TRF2 helps orchestrate a remarkable maneuver. The very end of the chromosome, which has a little single-stranded tail, is bent backwards. This tail then invades the double-stranded DNA further up the chromosome, prying the two strands apart and tucking itself inside, forming a stable lasso-like structure. This is called a T-loop. The dangerous raw end of the DNA is now completely buried within its own sequence, physically inaccessible. Even with the T-loop, there's a crucial point of vulnerability: the single-stranded DNA involved in the loop. This is where a specialized protein, POT1, which stands for Protection of Telomeres 1, does its most critical work. It binds directly and specifically to that single-stranded telomere sequence, like a hand gripping a rope. This grip does two things. It stabilizes the T-loop structure, and even more importantly, it physically blocks the activation sites for damage response proteins, particularly one called ATR. POT1 is the final do not touch sign. What happens if this system breaks? Scientists can run that exact experiment. In lab-grown cells, they can turn off the gene for a single shelterin protein, like TRF2. The result is immediate and devastating. The T-loops fall apart. The chromosome ends are exposed. The cell's repair machinery, seeing dozens of broken DNA strands, panics. It starts frantically stitching chromosome ends together. You get cells with multiple chromosomes fused into long, tangled chains. When that cell tries to divide, these chains are torn apart, shattering the genome.
Shelterin isn't just a physical shield. It's an active communication hub. It's constantly sending a signal to the rest of the cell that says, all is well. It does this by controlling access. It can recruit certain enzymes, like telomerase, telling it when and where to work. Simultaneously, it actively blocks other proteins, the DNA damage responders, from getting near. It's not a passive cap. It's a dynamic gatekeeper, a molecular switchboard routing signals to ensure the cell doesn't accidentally destroy itself.
So if we can just maintain this perfect cap, could we stop aging? Could we tell our cells to keep dividing forever? It's the ultimate biological question. The problem is that the telomere shortening that shelterin can't stop, the Hayflick limit, is a feature, not a bug. It's a built-in countdown that prevents a single cell from accumulating enough mutations over a lifetime to become cancerous. If you force telomeres to stay long and allow cells to divide indefinitely, you might not be buying youth, you might be opening the door to malignancy. The balance is extraordinarily delicate.
The system isn't designed for immortality. It's designed for stability across a planned lifespan. The Shelterin complex is the ultimate microscopic guardian, a machine of profound elegance that solves a problem most people never know they have. It performs an intricate molecular dance to hide our genetic endings from our own protective instincts. It is the protein that keeps our chromosomes, and therefore ourselves, intact, while simultaneously presiding over the quiet, inexorable ticking of our cellular clock.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.